structure of the Y chromosome. So here is a diagram representing the structure of the human Y chromosome. And you can see here that Y chromosome is divided into euchromatin and the heterochromatin. These are the two major regions of the chromosomes. So euchromatin is the active portion of the chromosome, while the heterochromatin is the inactive region of the chromosome, or you can say that heterochromatin basically is the condensed region of the chromosome, and mostly that is the inactive. Then a specific region of the Y chromosome is named as PAR, and that region is present on both the tips of the Y chromosome. You can see here that PAR is present on the upper tips on the short arm as well as on the tip of the long arm. So that PAR is basically an abbreviation of pseudoautosomal region. It means that is a specific region that is present on Y chromosome, but it also shares homology with the X chromosome. So pseudoautosomal region basically contain those genes that are also present on the X chromosome. So that's why it is named as pseudoautosomal region. As you know, autosomal chromosomes are same in male and female. So that's why this region is named as pseudoautosomal region, because this is also the region that is same in male and female, and you can say that is a region that is same in X or Y chromosome. Then a major portion of the Y chromosome is named as MSY. MSY is an abbreviation of male specific region of the Y. So MSY is basically that region of the Y chromosome that is specific to male and that is present only in the Y chromosome. So MSY, male specific region of the Y, it means MSY contain those genes that are playing an important role in the determination of male characters. Below the PAR region, there is a specific gene or the specific region of the chromosome, portion of the chromosome that is named as SRY. So SRY is an abbreviation of sex determining region of Y. So it means SRY is the most important portion of the Y chromosome that play an important role in the formation of male. So that is a general structure of the human Y chromosome. Before explaining the structure of the Y chromosome, I want to highlight some important factors about the sexual differentiation in human embryo. So how the gender decision takes place in human embryo. So you can see here, in case of female, the genotype should be XX. It means SRY is absent because SRY is a part of Y chromosome. So in that case, you can see here that when SRY is absent, then female development will start. And in that case, Mullerian duct will form the female reproductive tract, while the Wolffian duct will degenerate. So you can see here the formation of the ovaries, oviduct, vagina, uterus. So these all are the parts of the female reproductive tract. While when genotype is XY in the embryo, and that embryo is 5 to 6 weeks old, so gender decision takes place at that stage. So when genotype is XY, it means Y chromosome is present and that Y chromosome contains SRY. So in the presence of SRY, male reproductive organs will be formed. So you can see here the formation of the testis start, Wolffian duct forms the male reproductive system, while the Mullerian duct degenerates. So we have two ducts that are present in the embryo, and when embryo is present at its initial stages, then embryo behave as a hermaphrodite, means that embryo contain the gonadal ridges or the bipotential gonads, and these gonads has ability to either develop into male or to the female. So These bipotential gonads basically contain two layers. First is the cortex and the second layer is the medulla. So cortex layer has ability to form the female ovaries, while the medulla layer has ability to form the testes in case of male. Along with these bipotential gonads, two ducts are also present. One is the Mullerian duct and second is the Wolffian duct. So that is the embryo stage, or you can say that is the embryo at early developmental stages, and this embryo behave as a hermaphrodite. But later on, when development proceeds and the genotype is XX, in that case, SRY is absent and female reproductive system will be formed, while when SRY is present, then male reproductive system will be formed. So this picture is basically representing the importance of the SRY. So SRY is the most important region of the Y chromosome that contributes basically in the development of the male embryo. So what is the contribution of SRY? SRY is basically forming a specific product that is named as TDF. So TDF is a testis determining factor, and that is a product of SRY. As the name indicates, TDF is basically involved in the determination of testis. So it results in the formation of the testis, and how it forms the testis. Basically, TDF acts as a transcriptional factor, and you can also say that TDF is a master switch. So it further regulates genes involved in the development of the human embryo. So TDF activate a gene that is producing MIS or MIH. So TDF itself is a product of SRY, and TDF further act as a transcriptional factor or a regulating factor, and it controls further different, you can say different activity of different genes. So TDF further activate the gene, and that gene produce a product named as MIS or MIH. MIS or MIH is an abbreviation of Mullerian inhibiting substance or Mullerian inhibiting hormone. So when the level of MIH increases, it degenerates the Mullerian duct, and degeneration of Mullerian duct prevents formation of female reproductive tract. As I discussed in this previous slide, and you can see here that Mullerian duct is an important duct involved in the formation of female reproductive tract. So when SRY is present, that SRY basically produces the TDF, and TDF further regulates the gene that is involved in the inhibition of the Mullerian duct. So when Mullerian duct will be inhibited or Mullerian duct will be degenerated, in that case, female reproductive tract will not be formed. So obviously, male embryo will be formed. So what is the function of TDF? TDF acts as a transcriptional factor or a regulating factor that controls the genes involved in the formation of Mullerian inhibiting substance. That substance basically degenerates the Mullerian duct and prevents the formation of female reproductive tract. There are some autosomal genes that are also play an important role in sexual differentiation. One of them is the SOX4, other one is WT1, and both of them are present on chromosome number 11, basically act as oncogenes, but they also affect the kidney and the gonads. And as you can see in a previous slide, that biopotential gonads are basically associated with each kidney. So it means these two autosomal genes also affect the kidney as well as the gonads during the embryonic stages. Another autosomal gene is SF1, and that gene is basically participating in the steroid metabolism. And it was observed that the expression of this gene persists in males but extinguished in female. So during developmental stages, when the decision will be taking in case in favor of male, So the expression of SF1 persists in males but extinguished in case of female. So now moving towards the MSY, that is an important region of the Y chromosome, and 95% of the Y chromosome is MSY. So basically, David Page is a geneticist who first time discovered the structure of the MSY and explained that. So after 2003, when Human Genome Project was presented, then the structure of MSY was also described. So MSY basically consists of 23 megabyte, has three regions. First one is named as XTR, second is XDR, and third is the ampliconic region. So what is XTR? XTR is X transposed region. 15% of MSY is XTR, and that region shared 99% homology to the X chromosome Q arm 21 region. XQ21 represented the Q arm of the X, and on the Q arm, specific locus or specific region named as 21. So XTR basically share homology, or you can say 99% identical to the XQ21, and it only contained two genes. The next portion of the MSY is XDR, that is the X degenerative region, and it is 20% of MSY. It contains 27 single copy genes. It means these are the genes that are specific to XDR or specific to Y chromosome. That's why they are single copy genes, and it's they did not share any homology with the X chromosome. Another important region of the XDR contains pseudogenes, and these are the non-functional genes. And on the basis of these genes, that region is named as X degenerative region, because it was known that the pseudogenes may be functional in the past, but with the passage of time, they become degenerative or the non-functional. SRY, and that is an important region of the Y chromosome, an important gene of the Y chromosome, is also a part of XDR. The last region of the Y chromosome is ampliconic region, and it is the major region that contributes 30% to MSY. It contains 60 genes or transcriptional units. That 60 genes further divided into 9 gene families. Gene families basically contain a number of genes that share 98% homology. So, ampliconic region contains 60 genes that further divided into different gene families, and these gene families are 9 in number. And these gene families further contain 7 segments or amplicon. So, and what is the importance of ampliconic region? Basically, ampliconic region contain genes that are necessary for the development of the testes, function of the testes, and the male fertility. It means XDR contain the SRY, and SRY is involved in the formation of the testes. But further, the development of the testes and function of the testes and the male fertility is under the control of the ampliconic region. So if any mutation takes place in the genes of ampliconic region, it can cause the sterility in the males. So males become sterile. So ampliconic region is a major region of the MSY that contributes in the fertility of the males. So that picture is also describing the structure of the Y chromosome, and you can see here the X transposed region is represented with the pink color, and X degenerative region with yellow. Ampliconic region is represented with light blue color. Then here is a pseudo-autosomal region with the light green color, and then heterochromatin region with the red lines, and then some other regions are also present in the Y chromosome. So wasteland theory that was previously presented for the Y chromosome was rejected. According to the wasteland theory, Y chromosome is nothing, just a wasteland, contain very few genes that has no important contribution. But later on, after the discovery of the structure of the Y chromosome and importance of the Y chromosome, that theory was rejected. So that's all about the Y chromosome. the function of the SRY, importance of TDF, and the sexual differentiation in human embryo.